everyone. In this video, I am going to talk about inhibitors of glycolysis. Glycolysis is affected in the arsenate poisoning. What is this arsenate? It is the inorganic form of arsenic. There are two inorganic forms, arsenate and arsenite, and these are toxics. They are present in wood preservatives, insecticides, pesticides, fungicides, and even in the deep borewell groundwater. So what happens here? This is the step number 6 where glyceraldehyde 3-phosphate it is converted to 1,3-bisphosphoglycerate, and in this process there is formation of NADH, and this reaction is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, and here in this process one inorganic phosphate is added, and that's why there is formation of 1,3-bisphosphoglycerate. So how the arsenate interferes with this reaction? This arsenate, it is a structural analog of this phosphate, inorganic phosphate, and it competes with the phosphate for binding site on glyceraldehyde 3-phosphate dehydrogenase. So now instead of this inorganic phosphate, the arsenate will bind on this glyceraldehyde 3-phosphate dehydrogenase. And it leads to formation of 1-arseno-3-phosphoglycerate, releasing free arsenate to bind again with the enzyme. So this reaction continues, and thus it interferes with the formation of 1,3-bisphosphoglycerate. Then what happens? There is no production of ATP in this step due to arsenate binding, because there is no production of reducing equivalent, that is NADH. So this is how it interferes with the glycolysis. Now how this arsenate is different from arsenite? Arsenite is different, arsenate is different. So this arsenite acts differently. It binds to the sulfhydryl group of lipoic acid and inhibits various enzymes requiring lipoic acid. For example, pyruvate dehydrogenase complex, alpha ketoglutarate dehydrogenase, branch chain alpha ketoglutarate dehydrogenase. So these two are different, arsenate and arsenite. But this arsenate, it competes with the inorganic phosphate and it binds to glyceraldehyde 3-phosphate dehydrogenase, and it results into no production of ATP. Thus, it interferes with the process of glycolysis. The other inhibitor is iodoacetate. Iodoacetate is also potent inhibitor of glyceraldehyde 3-phosphate dehydrogenase, and it is an anti-glycolytic agent. Sodium fluoride is also another inhibitor of glycolysis. This sodium fluoride inhibits enolase. Now, what is the action of enolase? It converts 2-phosphoglycerate into phosphoenol pyruvate. But for the action of enolase, magnesium ion is required. Without magnesium ion, enolase cannot act. Now this sodium fluoride removes this magnesium ion and thus affects the activity of this enolase. So what happens? This fluoride removes magnesium and causes irreversible inhibition of enolase. Now why this is important? This is important in the estimation of plasma glucose. When we collect blood sample for plasma glucose estimation, we use this tube having grey colored cap, and this is also called as fluoride bulb. So what is there in this tube? There is sodium fluoride, which is anti-glycolytic agent, and potassium oxalate, which is anticoagulant, and it is present in the ratio of 1:3. And why we add sodium fluoride? Why we require sodium fluoride in this tube for plasma glucose estimation? Because if we don't add it, the glucose after collection undergoes glycolysis by RBC, so that the glucose value decrease by 5 to 10 milligram per deciliter per hour. So we will get then low false values, and that's why to maintain the blood glucose concentration in the blood, we need anti-glycolytic agent, that is sodium fluoride, which will prevent the glycolysis by RBC. So sodium fluoride is anti-glycolytic, and potassium oxalate, which is there in this tube, it is anticoagulant. In the next video, we will study about Rappaport-Lubrin cycle and its significance.